And let's go ahead and get started. And today we're taking a look at the human heart. So I have to apologize for what happened on Thursday. I got over the variable at 9 o'clock. It's just the nursing faculty and their students from one of the high school in Seymour came in at 10:30. We're supposed to be there at 9:30. And they didn't get done till after 11. So you know the result. I ended up having to record. So it was, I will say the presentation with the anatomage table was very impressive for the high school students. I wish they'd been there on time. So, oh, I recorded all that, didn't I? So I'm just gonna pretend like I never said it. Okay, but today since we finished up the blood pressure factors in blood vessels, we talked about resistance, we talked about blood flows through the various organs and stuff like that. Today, we're going to take a look at the number one pump in the world, our heart. Our heart is a structure that is about the size of our fist. And we should have it up now on the screen. There we go. How about, I love it when it works. Okay. So, yes, I got it now. Okay. So we are looking at the human heart. The human heart is about the size of our fist. This model, with the thymus gland still attached, is almost life size. You know, like I said, about the size of your fist. And it does, or I want to go ahead and pass, you get a good perspective on this. It is not the size of that big model I have up here. It's not the size of the cow hearts that we're going to see on Thursday for lab. Those things are the size of a football. You know, even this is about 3 times normal size. You wouldn't want to see a heart like this in a patient because it would indicate something's wrong. But the heart is unique among all the muscles. First of all, it's all cardiac muscle. That's why they call it the heart. But here, unlike in smooth muscle or skeletal muscle, where we can control either through the autonomic system or through the somatic muscle system, we can control the response to a stimulus. You know, how many muscle cells does it take to pick up a 5-pound weight? How many does it take to pick up a 10-pound weight? We can control the response. When we go to shake hands with somebody, we don't try to crush their hand usually. We control our actions. And not every muscle cell in an arm muscle is going to contract at the same time. You won't use all your cells in the biceps or the triceps or the deltoid to do something. In the smooth muscle, tells you the same thing. You know, how much are we going to contract? How much are we going to relax? But in the heart, when the heart is stimulated, which it is every 7/10 of a second, it will contract to its fullest extent every time. Every muscle cell will contract. And so the stimulus is a depolarization wave spreading across the heart. We're going to talk about that shortly. And it's going to cause a depolarization of calcium coming into the cell, not sodium, and potassium leaving. And that's going to generate a change, obviously, in the potential. And that's going to be our action potential spreading across the heart. And the heart's going to contract. And one of the things the heart does to make it even more efficient is, instead of depending on T tubules like we have in skeletal muscle. Everybody remember T tubules? So yes, those little things that go down into the muscle cells, you know, to extend the action potential down to open up the sarcoplasmic reticulum. We don't have T tubules. We have gap junctions. Those are holes in the cell membranes of the individual cardiac muscle cells. So the action potential doesn't have to come down from the top of the cell, but it shoots right through the middle of the cell. So our action potential spreads even faster. So the whole heart contracts together as one big cell instead of, you know, a few hundred cells here, a few hundred cells there. like in skeletal muscle, we're not going to, the cells that are contracting are all randomized around the muscle. So, but here, every muscle cell contracts and it pumps with the same force, as needed. So we increase the rate of contractions to pump more blood. We slow down the rate of contractions to pump less blood. We call the heart a functional syncytium. That means it acts as one big cell. So it contracts, it relaxes, it contracts, it relaxes, it does it together. And so we pump every 7/10 of a second, the heart contracts. We pump 70 beats a minute. And we do that for 80, 70, 80, 90, 100 years or more, over and over and over again. And in the heart, as I said, it's calcium, not sodium. Instead of sodium surrounding the outside of the muscle cells, it's going to be calcium. That becomes a really effective way of controlling hypertension because calcium comes into the muscle cells of the heart through calcium channels. And there are drugs called calcium channel blockers that can block the calcium channels. And if you do that, you slow down the heart rate, which lowers blood pressure. It's a very effective tool for controlling blood pressure, calcium channel blockers. So we use calcium instead of sodium. We still use potassium in here. The heart is packed full of mitochondria. It does not get its blood supply from what's inside the ventricles. That's a common, misconception. I guess that's the right way to say it. Where we think, well, there's lots of blood in the chambers. No, but we don't. We have our own coronary circulation. We have tremendous amount of mitochondria in the cardiac muscle. We have high oxygen demands in the heart. The heart is exclusively aerobic respiration. We don't have any glycolysis or very little glycolysis running in these cardiac muscle cells. Now, one advantage we have is that the myoglobin inside the heart, myoglobin, which makes the heart muscle red, because our heart is not bright red like this, but more of a darker red like the one there, depends on myoglobin. Myoglobin has the advantage of storing oxygen. So the cardiac muscle cell can be deprived of oxygen for up to an hour before it dies. And this is really important. I'm going to digress this for a second. The patient who has a myocardial infarction, that's a blockage in one of the coronary arteries. The muscle cells downstream of the blockage are going to die because they're getting cut off from their blood supply, but they're not going to die for an hour. So in that 60 minutes, you have time to intervene and put in a stent and open up that coronary artery so that blood can flow again. We'll talk more about stents, but a stent is essentially you run a mesh, sort of like a tent-like structure that's open. So making a terrible description of it. But you run that into the blocked coronary artery along with a balloon. You use the balloon to compress the blockage away from the center of the artery, and then you leave the stent in place to hold the artery open. And it's all done by running a long metal rod with a stent on the end of it up through one of the veins in the leg. It's as we say, minimally invasive, and I'll show you some more pictures of that later on. So that's a terrible description of a stent. Okay, now our heart, though, so we can survive a blockage, we have about an hour. Muscle cells are unique because muscle cells will last for hours. We know skeletal muscle will last for even more than an hour because rigor doesn't start for about 12 hours after death. Rigor requires us to deplete all the stored oxygen in the myoglobin. Well, in the heart, we've got an hour to play with. So you have a patient that's having a myocardial infarction, a heart attack. The first thing you're going to do when you get him into the emergency department is probably send him straight to your catheterization lab. the cath lab so that you can get a stent into them, or at least make sure that that's what's causing the chest pain. So the heart has its own electrical conduction system. It has its own nervous system, if you will. It is not dependent on any outside influence. The nervous system has no control over the heart. Now, the nervous system will help. The nervous system can do things for the heart, but the nervous system doesn't tell the heart when to work. The heart is what we say autorhythmic. It sets its own pace. It has what we call contractile tissue, which initiates a depolarization wave. So we have a series of contractile tissue that initiate a depolarization wave, or can initiate a depolarization wave embedded in the cardiac muscle itself. And we take advantage of that depolarization wave, the way it spreads across the heart and goes through the gap junctions, so that we get a constant response every time in a healthy heart. Our heart, by the way, contracts from the bottom up. The pointy end of the heart, the apex, is where the contraction wave starts contracting. Now, the depolarization wave spreads across the heart, gets to the pointy end of the heart, and then contracts upward. So our heart always contracts from the bottom up. Think of a water balloon. You'd think I'd play with water balloons like that I talk about, but I don't. Really, I don't. If you have a water balloon filled with water and you squeeze the top of it, and it's not tied off, a little bit of water is going to come out of the top, right? Everybody, okay. You've all seen water balloons at some point. If you squeeze from the bottom, though, what's going to happen? You're going to get more water, a lot more water, because you're forcing more water out. And that's why the heart contracts from the bottom up. So we have more blood coming out of the two big arteries. So we use gap junctions, and we use the contractile, the autorhythmic cells, we call them, to trigger the depolarization wave. And we don't just depend on one. We have 3 or 4 different types of autorhythmic cells that can take over if we have a problem with the first autorhythmic section. So we call these the pacemakers, the autorhythmic cells in the heart that will initiate the depolarization wave are the pacemakers. The entire wave of depolarization wave can go across the heart in a little more than 0.2 of a second. Now, remember, the entire cardiac cycle is 0.7 of a second. So in 0.2 of a second, our entire action potential can spread across the entire cardiac muscle of the heart. It's initiated in a structure known as the sinoatrial node. Sinoatrial node is in the upper back wall of the right atria. Here is your heart. There's your right ventricle, left ventricle, right atria, left atria. Your sinoatrial node, your first of the pacemakers, is right here in the back wall, the right atrium. Below that, we have the atrioventricular node. The atrioventricular node is here at the base of the atria, this region here, and is also an autorhythmic collection of cells that can trigger a depolarization wave. We have the atrioventricular bundle, which is the connection. This is the AV bundle, the connection between, the only connection between the electrical connection between the atria and the ventricles. Then we have what are known as the bundle branches that run down the middle of the septum. The septum is the divider in the ventricles. And then coming up from the base here, we have what are known as the Purkinje fibers. Each one of those areas I've highlighted can initiate a heartbeat. The sinoatrial node will initiate a contraction every 7/10 of a second. The atrioventricular node will give you, instead of 70 beats a minute, will give you about 50 beats a minute. The bundle will get you down less. And if you're depending on the bundle branches or the Purkinje fibers, you're somewhere around 30 beats a minute. It's survivable, but that's about all you can be doing is surviving. So take a closer look here. The SA node, the workhorse of the heart, if you will, it will depolarize every 7/10 of a second, initiating an action potential. It depolarizes faster than any of the cells around it. It will create a beat of about 75 beats a minute. We call this sinus rhythm, 70, 75, 72, somewhere in there. It would normally, if not controlled by the vagus nerve, generate a rate of 100 beats a minute. And this is what it would do. This is what it does. It would contract. It would depolarize itself so rapidly. If it was not controlled, we would have a rate of 100 beats a minute. But fortunately, the autonomic nervous system, which does not initiate our heart rate, but can control it, and speed it up or slow it down, uses the vagus nerve. The vagus nerve dumps right into the SA node. Here comes our vagus nerve right here. That's the vagus nerve. It's coming from the medulla oblongata, from the cardiac inhibitory center, and that dumps acetylcholine into the SA node. Acetylcholine is inhibitory in this case. It slows it down. So if the vagus nerve is working, it will dump acetylcholine there into the SA node and slow the heart rate down to 70 beats a minute, which is what we do all the time. Our normal heart rate is considered 70 to 75 beats a minute, thanks to the vagus nerve dumping acetylcholine there. If our vagus nerve were cut in an injury, which is entirely possible, our heart rate would accelerate to about 100 beats a minute. And that would be our normal resting rate. We probably wouldn't like that, but we could live with that. So, but however, if our vagus nerve is working and dumping acetylcholine in there, acetylcholine is inhibitory. It's a neurotransmitter. We all remember acetylcholine from skeletal muscle more than you ever want to hear about, I'm sure. We know that in skeletal muscle, acetylcholine is excitatory. Here, it's inhibitory. So, but the SA node will generate its depolarization wave, which will spread across both atria. This depolarization wave will spread across both atria like that. And when that depolarization wave reaches the AV node, the atrioventricular node, which is right here. My diagram's getting too messy. Here's an atria. Here's another atria. Right atria, left atria. SA node is up here. The AV node is right here up against the septum. The septum is a barrier. The septum is the wall of the right and left atria. We have a septum down here in the middle between the two ventricles, but the AV node is right here. Now our depolarization wave spreads across the atria like this. I don't think it's green, but and it will reach the AV node, and then it will move from there into the bundle, what we call the bundle of His. But before we go there, the purpose of the AV node is to slow things down. So we initiate our depolarization wave. The wave shoots across the atria. It gets to the AV node, but the AV node stops or slows down the depolarization wave by 1/10 of a second, which doesn't seem like a whole lot, but it slows it down by 1/10 of a second, because the reason we're sending a depolarization wave across the atria is we want them to contract. The atria do contract. They don't do much for us. They only top off the amount of blood in the ventricles by about 20%. Most of the blood ventricles are filling up faster. We'll get to that in a second. But we want to make sure that the atria have time to contract before the ventricles do, because the order of contraction is atria first, ventricles second. And so we slow down the depolarization wave by 1/10 of a second. That's what the AV node does. If the AV node were running by itself, if there was no SA node functioning, the AV node would still give us 50 beats a minute. Entirely survivable. Pretty much you could do almost everything that you could do before, except maybe run up a flight of stairs or exercise vigorously. 50 beats a minute is a very survivable rate. But it also means there's something, if the AV nodes are doing it and not the SA node, it means there's something wrong up here at the SA node. So, question.
So let's go to the pacemaker.
That's exactly what a pacemaker does for us. The SA node is our body's natural pacemaker. So is the AV node. An artificial pacemaker would be implanted to do the same thing that the SA node does, to stimulate an action potential every 7/10 of a second. And so it's designed to stimulate the muscles right in that same location. You would typically plant it off to the side somewhere with its battery, and then it'd be hardwired into the back wall of the right atria. In lieu of that, before that was done, you'd be depending on the AV node giving you 50 beats a minute, which, if you were used to running at 75 beats a minute and you were cut back to 50, you'd probably notice that, but it's not going to be imminently fatal or anything like that. It's very survivable at 50 beats a minute. Now, the AV bundle, or what we call the bundle of His, is located directly below the AV node, and it's the bundle of His is right here. The AV bundle is in the septum at the top of the ventricles. Now here's our divider between the two ventricles. This is the septum right here. Here's the left ventricle, right ventricle. And in between is a thick layer of muscle tissue. That's the septum. What we call it the interventricular septum. And right in this region here is the bundle of His. It is the only electrical connection between the atria and the ventricles. The only communication, the only way an action potential gets into the ventricles is through the bundle of His. So we go from the SA node, a depolarization spreads across the atria. It gets down to the AV node. It slows down for 1/10 of a second to give the atria time to contract before the ventricles do. And then the depolarization moves into the bundle of His. It has now jumped from the atria to the ventricles. And from there, it will go down the middle of the septum. Down the middle of the septum are what we call the bundle branches. So here's our septum. right down here. You know, our bundle, this is up here. The bundle branches run down here to the apex, or as refer to it as the pointy end of the heart. So they carry the bundle branches, carry the depolarization wave down the middle of the septum. This is the septum right here. I want to call it IV septum. I don't want to write interventricular again. And so our depolarization wave travels down here to the apex. And we have one last stop at the very bottom of the heart. At the end of the point, we have coming up in the walls of the ventricle. This is a ventricle. This is the right ventricle. Here is the left ventricle. Here, the Purkinje fibers are going to come from the apex and come up like this. Purkinje fibers. They innervate the cardiac muscle on either side. So if you were to take a look at a heart, it would look something like this. Maybe I can get this camera to work. There we go. Here is the apex of the heart is right here. And these are the Purkinje fibers in white coming up on both sides from the apex. The apex is here, the pointy end, and see if better. There we go. There's the apex right here, and these Purkinje fibers come up on both sides. This is all cardiac muscle here and here. The bundle of His was somewhere up inside here, because here's the septum right there. And so the signal comes down the septum, which is that's the picture. There's our, there we go. Here's our septum. There are the Purkinje fibers here and here, but down the middle of this are the bundle branches. And we're going to send our signal down the bundle branches. We're going to send our signal to the apex and then up the Purkinje fibers, and they will initiate depolarization and contraction of the cardiac muscle cells from the bottom of the ventricles up. So our squeezing action, our compressive action starts bottom up to force out blood through the two large arteries that are leaving the heart. So everything is, it is sequential. We go SA node to AV node to bundle of His to bundle branches to Purkinje fibers, and we contract upward. If the bundle of His is the only structure working, if the atria aren't contracting, if there is no SA node function, if there is no AV node function, the bundle of His can take over and trigger a contraction rate of about 30 beats a minute. Survivable, but barely. So the heart's beating at 30 beats a minute. It may or may not be adequate to send blood to every organ like it should. So from the time it starts to the time it's over, the time we have contraction, from the time we initiate the depolarization from the SA node to the time the heart contracts vigorously is 0.2 seconds, 2.22. So 2/10 of a second is the contraction phase of the cardiac cycle. Remember, the cardiac cycle is 7/10 of a second. The rest of the cardiac cycle is spent in diastole. It's in rest, because this is when, once the heart contracts, now it's going to go into its rest phase and fill back up with blood. And even that's not a correct statement. It only pumps out about half the blood in the ventricles every contraction. So when I say fill back up with blood, it's only going to replace the blood that was pumped out. So this is our intrinsic cardiac system. Intrinsic means it's built in. We have no outside influence. The best that the autonomic nervous system can do is slow down the heart rate by dumping acetylcholine into the SA node, or speeding up the heart rate by dumping epinephrine into the SA node and into the myocardium. Because there are times when we want to speed up our heart rate. You're running up a flight of stairs. You're running to your car. You're just running, and your heart, you're working out, or you're lifting. You're building something, and you're lifting heavy objects, and your heart is meeting the demands of your tissues, and it is beating faster. The heart rate goes up as there's more demand for oxygen. So the autonomic nervous system, which is aware of this demand, will stimulate the heart to beat faster. And then when you're done, the autonomic nervous system slows your heart back down to your resting phase. You remember, the parasympathetic system is your rest, digest, and defecate. Your heart rate slows down. You're relaxed. Acetylcholine is the dominant neurotransmitter, and it slows your heart down. When you're resting. And then in a fight or flight or exercise stage, you're busy dumping epinephrine into the heart's beating faster. But the heart will continue to contract typically every 7/10 of a second on its own without any outside influence. That's why we say it's intrinsic. So the sinoatrial node will give you about 70 to 75 beats a minute, or 7 to 75 contractions. Contraction of the heart is not the same as the heartbeat, because the heartbeat is the sound of the valves closing. We'll get into that in a second, but let's just say 75 beats a minute. 50 beats a minute is the AV node. The bundle of pits, the bundle of branches, and the Purkinje fibers, which come up here from the apex, 30 beats a minute. Survivable, but barely. So, questions on this? Lots of individuals that have problems with the conduction of the system in their heart, you can often identify from the electrocardiograph where the problem is. If the AV node is not firing properly, if the SA node is not firing properly, if there are blockages, literal blockages in the bundle branches, we call it heart block. And we can see a change on the electrocardiograph based on that. So a relatively easy, non-invasive technique works very well to give you a good idea, because our heart is consistent. When there is a malfunction in one of these nodes, or there's a blockage somewhere in the conduction system, we can see the results on the electrocardiograph. So, problems. Lots of things can go wrong. Arrhythmias are an irregular heartbeat. Where the heart is, one of the chambers, or both, the upper chambers or the lower chambers are beating out of sync. The atria, one of the most common problems we have, one of the most common arrhythmias is what's known as atrial fibrillation, where the atria quiver out of sync. It's a very common arrhythmia. An arrhythmia is an irregular heart rate. Now, atrial fibrillation is not the same as ventricular fibrillation. Atrial fibrillation, many people have it. Many people find it very annoying or uncomfortable, or maybe even a little scary. But what happens here is the atria are not able to contract and dump their blood into the ventricles, but the ventricles don't really depend that much on the atria. The atria only add the final 20% of blood to the ventricles themselves. So the rest of the blood that's flowing into the ventricles is all passive. When the heart's at rest, the valves between the atria and the ventricles are wide open. Blood's flowing in passively. The ventricles are filling back up. And if the atria can't contract, okay, the atria can't contract. You have less blood in the ventricles, but the ventricles will compensate. So is it a long-term problem? Yes, it can be very much of a long-term problem. In some cases, atrial fibrillation can lead to uncontrolled contraction of the ventricles, which is going to cause death in about 5 minutes. Anyway, fibrillation is a bad word. It's uncontrolled contraction, whether it is ventricular fibrillation, which means that you have to reset the overall rhythm of the heart, or atrial fibrillation, which in many cases will, initiate and then stop and go back to a normal rhythm. So, okay. If the ventricles are in fibrillation, then you have to use one of those AEDs. You have to shock the individual to shock the heart back to its normal rhythm. Yeah. Pretty extreme treatment, but it works. Okay. Sometimes different things can go wrong with the SA node. If the SA node malfunctions, for whatever reason, if the SA node slows down, essentially the autorhythmic tissue that has the highest rate will take over. So if the SA node were to malfunction and drop below 70 beats a minute, 60 beats a minute, then the AV node will take over. The autorhythmic tissue, the pacemaker tissue with the highest rate will always be the one to take over. So maybe the SA node doesn't work right, the AV node will work. Sometimes the SA node will have what's known as it will trigger a premature contraction, where you're running along and your sinus rhythm on the EKG, you have your P wave, which corresponds to atrial contraction, your QRS, and we're going to get into all this in a little bit here, your QRS complex, which is the ventricular contraction, and your T wave, which is the relaxation of the ventricles. And somewhere along in the EKG, all of a sudden you get this big spike of a contraction. Where did that come from? We didn't want to see that. Because sometimes if the SA node is malfunctioning, it can trigger an extra contraction in there. And so the ventricles contract before they've had time to fill up to reload for the next normal contraction. And so what will happen is you'll often have a delay in the cycle. You have even more blood in the heart. When the heart contracts, you hear this thudding sound, simply because we had an extra contraction in there. So, yeah. It says excessive caffeine. I don't think there is such a thing as excessive caffeine. I think that's a misprint. So 12 cups a day is just a good start, but that's neither here nor there. But yeah.
So if the SA node causes PACs, does AV node cause PVCs? Because they're different, they look way different.
Oh, they do. They do. The AV node would have to override the depolarization wave, because normally the AV node isn't, you would have to have a failure of the SA node for the AV node to be the initiator and trigger that.
So the SA node causing PVCs also?
They can.
Okay.
So you can, because it'll trigger what we call an extrasystole. So just about anything can happen, basically. But because if the SA node is functioning, the AV node should not be doing anything. The AV node should simply be let's slow things down by 1/10 of a second. But it can, in lieu of the right signal, the depolarization wave, take over. Now, is it possible for it to create its own depolar? Yeah, anything's possible. And so you'll see all sorts of irregularities on the EKG. So yeah, long answer.
Good answer.
Okay, thank you. So Okay. Now, other problems. If the AV node, if the SA node's working fine, initiating a depolarization wave, and the AV node isn't working, then we create what's known as heart block, where we are not sending the signals because the AV node slows down the depolarization wave. It directs it through the bundle of His, which goes down the bundle branches in the middle of the heart. If the AV node isn't functioning, then we're going to block those signals. And so it slows down the heart rate below the level of adequate circulation. Yeah, so we say we have a heart block in the patient. Now, the heart block from the AV node or from the bundle of His, something is the signal is not getting down the middle of the heart in the septum, the interventricular septum, the part here in the middle. And so the Purkinje fibers are not getting the signal to trigger contraction of the cardiac muscle. So that can happen too. And so you have to, like this requires the use of a pacemaker because we have to be able to get the signal from the atria through the inner, there's another septum, the septum between the atria and the ventricles, through that into the interventricular septum to stimulate the Purkinje fibers. So all sorts of things that can go on. It's amazing that everything works so well, so long for us. Okay, what about the normal intrinsic rhythm of the heart? Well, as I mentioned earlier, the autonomic nervous system, using the medulla oblongata here in our brainstem, the medulla has a cardiac acceleratory center and a cardiac inhibitory center. The cardiac acceleratory center uses epinephrine. That's not the 12th cup, by the way. It uses the cardiac nerve to send signals to the SA node, to the AV node, and to the myocardium, the cardiac muscle, and telling it to contract faster. So the cardiac acceleratory center does exactly what it says its name is. It tells the heart to speed up and uses epinephrine. Well, we don't do that very often. We only do that when we're stressed, when we're in a fight or flight, or when we have greater demands on our muscle cells. Our cells need more oxygen and need to get rid of more CO2, which is probably the bigger driver. We need to get more glucose out to the cells, so we have to pump the blood faster. We have to have a higher heart rate. Autonomic nervous system, which is aware of all these needs, stimulates the release of epinephrine, and the heart contracts faster. On the other hand, the inhibitory center, parasympathetic side, vagus nerve. Vagus nerve slows down the heart rate with acetylcholine. It dumps acetylcholine into the SA node, into the AV node, and we slow down. We normally maintain at 70 beats a minute or 75 beats a minute, but we can drop down when we're at rest, below 70, and that's perfectly fine. When we're sleeping, we don't need to have our heart rate running at 80 beats or 75 beats a minute while we're sleeping. So we depress the SA node and the AV node with the acetylcholine released by the vagus nerve coming from the cardiac inhibitory center in the medulla. So the medulla oblongata in our brainstem has both a cardiac acceleratory and a cardiac inhibitory center. They probably are located side by side. The cardiac nerve and the vagus nerve probably run pretty close to each other because that's just what they do. And one's dumping out acetylcholine to slow things down. The other one's dumping out epinephrine to speed things up. And it just depends on the situation. The autonomic nervous system is giving us a comprehensive picture. And it looks something like this. You can see the medulla up there at the very top. we have the inhibitory center and the acceleratory center. The inhibitory center is showing the vagus nerve in purple coming down to the SA node and AV node, dumping acetylcholine in, slowing down the heart rate. Here we have the acceleratory center releasing norepinephrine into the SA node, it's the green one, SA node, AV node, and into the muscle area themselves. So we can speed up, we can tell the muscle to accelerate, to contract faster. It's more receptive to an additional, a faster rate of depolarization. So, and we don't have to do anything about it. It happens automatically because it's part of the autonomic nervous system. We slow down, we speed up, we slow down, we speed up, we meet the demands of living, whatever they are for us. In a fight or flight response, when we're dumping a whole lot of epinephrine into our system, our heart's going to speed up and pump blood faster to go to the brain, the heart, the brain, the lungs, and the muscles, and to itself at a higher rate because it's a fight or flight. We're doing something in fight or flight. Questions on any of this? Okay, now I mentioned this electrocardiography. We call it technically it would be an ECG. Most of us call it EKG from the German version where it's spelled with a K. It is a graphic display of the summary of all the action potentials going through the heart at one time. So instead of trying to see what's happening at the SA node or the AV node, we get the EKG machine will interpret everything and give you a display of, this is what we call sinus rhythm. This is the heart reacting. So typically when you have an EKG, you're going to have what we call leads. If you've ever had one, you know what I'm talking about. But they take like a little electrode, and they put them on your wrist, they put them on your ankles, they spread them across your chest, and they're going to pick up the electrical signal as it spreads across the heart, and from the extremities, and then put it all together in one signal. And so what it looks like is this. This is a typical EKG. We have a series of depolarization waves that show up. The first one is called the P wave. P wave over here is the depolarization of the SA node and the atria. So the P wave is what we call atrial contraction. So when we see a P wave, we know the atria have depolarized, and if they depolarize, they're going to contract. So we don't see a contraction wave. We see that, we're not looking at the movement of the muscle, we're looking at the electrical signal. The P wave is the depolarization of the atria, meaning that they're going to contract. Now, right next to that, we have a section called the QRS complex. See the Q, the R, and the S. This is going to be ventricular depolarization. The ventricles are going to depolarize and contract. Again, we don't see the contraction of the ventricles. What we see is the depolarization wave, and we know that when they depolarize, they will contract. At the same time, if a muscle cell depolarizes, it has to repolarize. Well, repolarization of the atria occurs during the QRS. The QRS is such a powerful action potential, depolarization wave, if you will, that it hides the sign of the atria repolarizing. Because we depolarize and then we repolarize. So what we have here is we have the P wave, but we don't see any repolarization of the atria. But it's happening. The signal is totally blocked by the QRS complex, because that is the contraction of the ventricles. That's a big, powerful contraction. The atria are like, I'm going to give my contraction now, and then the ventricles are like, woof, I don't think it makes that sound, but you know what I mean. Okay. But since the atria are already done and the ventricles have already contracted, they still have to repolarize. And what we see here is what's called the T wave. The T wave is the repolarization of the ventricles where calcium leaves the cell, goes back, you know, where we change it, you know, potassium's outside, calcium's inside, and then the sodium, in this case, the calcium potassium pumps are going to pump everything out. And, you know, once we have gone through the QRS complex, we start the process of diastole. Because as soon as we've contracted, as soon as we see the QRS complex, we know the ventricles have contracted and blood has left the heart. And as soon as that happens, we start diastole. You know, diastole starts at 0.22 seconds after the SA node fires. You know, when the SA node fires, you get your depolarization wave. It spreads across the atria, goes down through the bundle branches to the Purkinje fibers, up the Purkinje fibers, and then we contract from the bottom up. And as soon as we contract, we're going into rest. So the next, you know, remember it's a 7/10 of a second cycle. So for the next half a second, more or less, we're going to be at rest and we're going to be filling the ventricles back up again. So the T wave is the repolarization of the ventricles. And over here is this little U wave. We don't often see that. But remember, the Purkinje fibers had to contract too to get the muscles to contract. The U wave, if it's seen, is the repolarization of the Purkinje fibers. And they're fine because it's, you know, the U waves, you know, at the very end of the EKG here, the heart's at rest. You know, as long as we're, we may not even see the U wave, but if we do, it's just showing that the Purkinje fibers are repolarizing back to their normal resting state. And so this is what the electrocardiograph looks like. We have a P wave for atrial depolarization, meaning the atria have contracted, followed by a QRS complex, meaning the ventricles have depolarized and they're contracting, followed by a T wave where the ventricles are repolarizing. We may see a U wave, we likely won't, but if we do, it's the Purkinje fibers repolarizing. And we don't see anything from the P wave because their repolarization is taking place in the middle of the QRS. It's lost. The signal is just blocked. Problems that the EKG can detect. Well, all sorts of things. The rate of the EKG, the depression of the waves or the elevation of the wave can indicate all sorts of things. In the QRS complex, for example, the R, the middle deflection, it's QRS. In the middle of that, if the R wave is elevated, it can mean there's a problem with the ventricles. The depolarization of the ventricles, for whatever reason, why is the R wave? There's your R wave there. Why is the R wave elevated? Shouldn't be, that should be the normal, deflection of what they call the R wave. It shouldn't be an elevated wave. What about the ST segment? The ST segment is this area from the S, the bottom of the QRS, over to the beginning of the T wave. That's what it's highlighted there as the ST segment. The ST segment if the ST segment is elevated or depressed, sometimes it can have a spike right there at the ST segment, or it can be inverted at the ST segment. That can mean that you're not getting adequate perfusion of blood into the cardiac muscle. It may mean ischemia. Ischemia, the term ischemia means you're not providing enough blood to the tissues as needed. So the tissues are starving in here. That's a possibility. We may have if the QT segment, let's go back here again, the QT, Q goes from here to here. If this segment is excessive, now remember, you've got the electrodes on your patient and you're just looking at this chart, you're not doing anything invasive. But if the QT segment is long, it means there's a problem with repolarization of the ventricles. And so by looking at this EKG, sometimes you can see the QRS is inverted. Sometimes the T wave is inverted. There are all sorts of things here that these deflection waves, that's what we call them, can tell you what's going on in your patient's heart. There are all, blocks or fibrillation, some heart flutter, any of these are going to show up, as unique and repeatable changes on the EKG. The transient anomaly that happens once and goes away may or may not be of concern, but a continued, anomaly of like an inverted T wave again and again and again, or an extra long time between the P wave and the QRS. The atria contract, and then it takes a long time for the ventricles to contract. What does that mean? What's causing that? What's affecting the depolarization wave spreading across the heart? That your atria will contract, and then there's this gap before the ventricles do. Things like that are going to show up. Okay, here we see top. This is what we would call normal sinus rhythm. You can see the P wave. This is the P wave right there, QRS, and there's the T wave. Again, P wave right there, QRS, T wave, P wave, QRS, T wave, T wave, QRS, T wave. Not seeing the U wave on here, and that's okay. On the middle strip, we see this squiggly line in here. See all that, you see a normal QRS, and you see a T wave, and then you see these squiggles in here. The squiggles indicate atrial fibrillation. In atrial fibrillation, the atria are continually depolarizing and quivering. They're not effective, in any kind of contraction. Well, atrial fibrillation is not life-threatening immediately. It may never be life-threatening, because the atria, as we're going to see later on, really do most of their work by not doing anything. So, but this can be a concern. There does seem to be some delays in here, between your depolarization of the ventricles here, and there's a extra long gap right here, because we're seeing more time in between the contraction of the ventricles. Not a good thing either. And of course, this one at the bottom is what we call ventricular fibrillation, and that's never good. Because instead of contracting instead of a squiggly line like we get when the atria are fibrillating means to contract uncontrollably. The ventricles in atrial fibrillation are still functioning. Your patient might not even be aware that they're in atrial fibrillation, in AFib, as they say, because it can be, they feel fine, So, but here, ventricular fibrillation means that the ventricles are not contracting. There is no rhythm. There's no sinus rhythm here. The ventricles are contracting so rapidly, they're not getting time to fill up with blood to send it to the body. So your patient is being deprived of oxygen. This is going to cause brain death in about 5 minutes. Heart muscle is going to be fine for about an hour. But this is going to cause brain death because you're not effectively pumping blood. You shouldn't have this many contractions. Consider the rate even at AFib. You have this much time interval, between contractions here, and the contractions are piling up on top of each other. And we don't want that, because that will kill your patient literally in about 5 minutes, because the brain's deprived of oxygen. Questions on this? Or is it just Monday morning? So, okay, let's talk about the cardiac cycle. Cardiac cycle is broken down into the systolic and the diastolic phase. The entire cardiac cycle are the events that happen in that 7/10 of a second time phrase for the heart, where we have contraction of the heart and relaxation of the heart. And we spend much more time in the diastolic relaxation phase than we do in the contraction phase. Now, the cardiac cycle, every part of the heart goes through this contraction and relaxation. Atria will contract and pump blood into the ventricles. Then they will relax. The ventricles will pump blood to the body or to the lungs, and then they will relax. So everybody goes through systole and diastole. Now, there are 4 parts to the cardiac cycle that incorporate the systolic or systole and diastolic sections. Ventricular filling is exactly what it sounds like. The ventricles fill up with blood, getting ready for the next contraction. Then you have isovolumetric contraction, where the ventricles become sealed chambers and start squeezing. To get blood to come out through the arteries, through the aorta, and through the pulmonary trunk, you have to raise pressure of the blood in the ventricles higher than the pressure of the blood in the aorta and higher than the pressure of the blood in the pulmonary trunk. Once the pressure in the ventricles, as they're squeezing themselves, rises, then the valves that control flow out of the ventricles will open up and blood will shoot out to the lungs or to the body. That's the ventricular ejection phase. We've raised the pressure inside the ventricles high enough, we're squeezing that water balloon enough to shoot blood out through the pulmonary trunk to the lungs and the aorta to the body. Different pressures, so. And then we go, as soon as we eject that blood, we're going into the relaxation phase. Ejection of the blood takes place at the 0.22 seconds, 2/10 of a second into the cardiac cycle. We've ejected the blood, and now the next 5/10 of a second, a half a second, we're going to be in the rest phase to reload the cardiac muscle, reload the ventricles. So, ventricular filling. Here we are, the atrioventricular valves. The valves between the atria and the ventricles are open. And so, here's our heart, more or less. The atrioventricular valves are here and here. There's the septum. There's the size of our, this is the left ventricle, this is the right ventricle. And this is the tricuspid valve here. And here is the bicuspid or mitral valve here. And these are the AV valves, atrioventricular valves. They are the 2 valves that connect the atria to the ventricles. In ventricular filling, these valves are open. Blood is flowing in passively into the heart. Over here we have the superior vena cava, Superior vena cava is draining the top 1/3 of the body. So stuffing into the right atria. Here we have the inferior vena cava, the lower 2/3 of the body. Blood coming up here into the right atria, blood's coming into here in the right atria. On the left side, we have the pulmonary veins. We have 4 pulmonary veins here, 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 and here. Pulmonary veins. Blood's coming back from the lungs. High O2. Blood's coming in here and here and here and here. So the right atria and the left atria are receiving blood under low pressure. Because the return flow, well, that's messy. Return flow to the right side of the heart is under very low pressure, 0 to 10 millimeters of mercury. Return flow of the highly enriched oxygenated blood coming in on the left side from the lungs is also under very low pressure, 0 to 10. Remember, venous flow is always very low pressure. Veins always return blood to the heart. It doesn't matter if they're lower in oxygen or higher in oxygen. The atria are receiving this blood. The pressure in the atria is very low, but it is greater than what's in the ventricle. So the ventricle pressure is even lower. So blood flows into the atria and into the ventricles. Let's see if I can get this camera to work again. Here, okay, here is the left, this is the left atria right in here. This is the mitral valve right here. These are 2 of the pulmonary veins here, and you can see the other 2 over here. They run across the back of the heart. So anyway, so here is our left atria with 2 of the pulmonary veins. Here is our mitral valve here. Blood's coming in passively into the atria and going right through this valve into the left ventricle down here. This is the left ventricle. So, you know, blood's coming in, these valves are wide open, everybody's at rest. Over on the right side, better. Okay. There we go. Sort of. This is the right atria. We have in the right atria, we have down below here, we have the tricuspid valve right there. And we have these red circles. And up here at the top of right there is the superior vena cava, superior because it's on top. And you can't really see it very well. But if you look at the bottom here, there's the inferior vena cava bringing up blood from the bottom of the heart, the bottom of the body. So both sides are receiving blood. The right atria is receiving blood from the superior vena cava and the inferior vena cava, all passive, all low pressure. This blood is lower in oxygen. That blood has to go to the lungs to become reloaded, become more oxygenated, bring it back up to 100%. The left atria is receiving that oxygenated blood, that 100% oxygenated blood, coming in passively. Again, very, very low pressure. And it's passing through the mitral valve down in the left ventricle. So the right side is receiving blood to pump to the lungs, and it's filling up. The left side is receiving blood that's already been to the lungs and pumped to the body. Remember, the heart is 2 independent pumps. Okay. Everybody is in diastole. Everybody's at rest. Blood's flowing in passively. Now, we are in this diastole phase, and when we get to about 80% full, when the ventricles get to about 80% full, then that's when the SA node will fire. The SA node will fire, and all it will do is top off the ventricles. See, the ventricles are going to be I have one more board space on. Okay. Here's our heart. Here, atria. Right ventricle, left ventricle. By the way, the amount of space in here corresponds to the size of the ventricle chambers too, because the left ventricle is much larger than the right ventricle. As this blood flows in here passively through these valves in here and in here, it's going to fill up. When it gets to about 80% full, that's the trigger for the SA node fires at 80% full. The atria contract and top off the remaining 20% of blood. And they don't measure, oh, I only got 19%, I only got 18%. It's about 20%. So how much do you end up with? Well, the ventricles, both left and right ventricles, will have about 130 mils of blood in each chamber. Okay. And that's fine. They can be a little bit off. What's critical, we'll get to that in a few minutes here, is the amount that's ejected has to be the same. So we top off the ventricles, everybody's still at rest except for the atria. The atria have done their thing. They have depolarized. You've had your P wave, and blood has been ejected gently into the ventricles and filled them up to their full 130 mil volume. And the atria are done. They go back into their rest phase. They've contracted, however short a period of time, and that 2/10 of contraction phase is. They've contracted. We're done. And now we go into ventricular ejection, or rather isovolumetric contraction. So what's going to happen now? Well, before we go any further, we contract, the AV valves are contracted, they filled up the ventricles. Now, the heart valves have to go shut. We had passive filling in here. And this is fine. Everything's good. We filled up, atria have filled up, the atria have contracted. We may or may not hear any sounds for that, but not normally, not usually. And now that our ventricles are completely full, we're getting ready for the heartbeat. The heartbeat is the sound of the AV valves going shut. So before the blood can be pumped, we have to start squeezing the ventricles. The QRS complex in the EKG triggers that's the depolarization of the ventricles, of the heart muscle. The ventricles start squeezing. At the same time, we can't have the valves open. We can't have the tricuspid valve or the mitral valve open here, because if they were open, then when we contraction, we just shoot blood back up into the right atria and the left atria. So we have to close the valves. And this gives us our heart sounds. The sounds of the heart, sounds of the heartbeat. The first heart sound is a low-pitched lub sound. So we get this lub. And the lub is the sound of the tricuspid and mitral valve being slammed shut. The chordae tendineae, these little parachute lines that we see here on our on our valve, these little white lines here, these little white structures here, the chordae tendineae here and over here, they are attached to the valve flaps. The valve flaps then, when as the heart is ready to start contracting the ventricles, the muscles that are attached to the chordae tendineae will pull on those strings. And when they do that, then the valve flaps will be pulled shut. And so the tricuspid valve shuts and the mitral valve shuts at the same time. The mitral valve tends to shut slightly before the tricuspid, but what we hear is a low sound. The valves are shut. And what that does is that gives us a sealed chamber then. Here, the tricuspid valve has gone shut. We can squeeze the right ventricle. The mitral valve is shut. We can squeeze the left ventricle. And blood's not going to shoot back up. And here we now have a completely sealed chamber. No more blood is coming in from the atria at this point. And we're going to squeeze and squeeze and squeeze. We're going to raise the pressure inside the ventricles. Now, the problem here is that we have two large vessels. This blue one here is called the pulmonary trunk. The red one is the aorta. There is back pressure in both of those arteries. The pulmonary trunk has blood inside of it. There is another valve here called the semilunar valve. The semilunar valve keeps the pulmonary trunk shut, so blood doesn't flow back into the heart. There's a certain amount of pressure on the other side of the valve. So we have to raise the pressure in the right ventricle here to exceed the pressure in the pulmonary trunk. Pulmonary trunk leads to the two pulmonary arteries. So to get blood to leave the right ventricle, we have to raise the pressure here to exceed the pressure here. Well, the pressure here is usually between 10 or 15 millimeters of mercury. And so if we get up to 20 millimeters, we can force that semilunar valve open, it flips up, and blood shoots out into the pulmonary trunk. Over here, a little bit different. Pressure in the aorta, the largest vessel in the body, the one that takes blood to every part of our body, is here. The pressure in there is usually around 120 millimeters of mercury. So we have to raise the pressure here high enough to exceed that. So it's 100 in here, we have to get to 120. If it's 120, we should get to 140. Once we raise the pressure in the left ventricle greater than the mitral valve, then I'm sorry, the semilunar valve here, the aortic semilunar, then blood will shoot up into the aorta. And it's only if we raise that pressure. So during the contraction phase, we are squeezing the blood, the muscles are contracting, we're squeezing the blood. It's getting more and more powerful. We're raising the pressure inside each ventricle. When we exceed the pressure on the other side of what we call the semilunar valves, there's a semilunar valve at right here. This is the pulmonary semilunar. Here's the aortic semilunar. Once we exceed the pressure on the other side of the semilunars, blood's going to shoot through those valves and go out to the body. And we call that ventricular ejection. So the tricuspid and mitral valve are shut. The semilunar valves are open. And they'll stay open as long as there's high pressure blood or higher pressure blood leaving the ventricles. At some point, the pressure on blood in here starts to drop. We have it, we've contracted, we've pushed that blood out, and then the pressure out here in the pulmonary trunk starts to drop. And when that pressure drops, the blood drops back against the semilunars. The semilunars are passive structures, they are cup-shaped, so that when blood hits them, they go shut. The same thing over here, the aortic semilunar. As soon as the pressure starts dropping, and the blood will slam against the aortic semilunar and we close it off. And that gives us the second heart sound. The dup sound, our heartbeat is a lub dup. The lub is the sound of the AV valves, the tricuspid and mitral going shut. The dup is the sound of the semilunars going shut after blood has left the heart. Its pressure has dropped a little bit and it comes back up against these valves. And so we get the second heart sound. So now we have the pulmonary semilunar closed, the aortic semilunar closed, and the we have more and more. We're still closed here. We're still closed at the mitral and the tricuspid, but blood continues. Blood is continuing to return to the heart the whole time this is happening. Blood's continuing to return to the heart, and blood's filling up the atria, raising the pressure in the atria, which will eventually force open, cause these valves to relax and let blood come in here. And then we go into isovolumetric relaxation, which is where we're going to stay for about a half a second, 0.5 of a second. The ventricles relax, the semilunars are closed, blood flows in passively into the ventricles all over again. We may even see a change in pressure as the semilunar valves close and blood slams up against it as it's bouncing off there. We may get a temporary, transient rise in blood pressure called the dicrotic notch, and it goes away as fast as it happens. So that is isovolumetric relaxation. So the entire cycle is 0.7 of a second, up until the point of when isovolumetric relaxation starts. We're only 0.22 seconds into it. So 0.2 of a second in, we go into relaxation phase, and we stay there for about 0.5, a half a second, which is a very long time as far as the heart's concerned. And that gives us our relaxation phase, and that's where we are as we go into ventricular filling again. Everybody's at rest, and we start the whole cycle all over again. So, questions on any of that? Well, let's take a little break, and we'll come back and talk about cardiac output.
I was gonna ask you, so why do babies have like such a higher heart rate than adults?
They have a higher demand for oxygen. They have a higher demand to get rid of their waste products. So, and it will come down.
Yeah.
But initially, they are pumping blood as fast as they can throughout their body.
Yes. My baby, I got her that outlet, and then it's like at night it's like 160, and she's just asleep.
Oh yeah, yeah.
It's crazy.
It's perfectly normal.
Yeah. Because I read about it, and it's like if it goes under 80, it like sets an alarm off that is like that they're not getting enough oxygen.
Right.
So I'm like, that's crazy.
Well, they have high demand because they remember they're going to grow like crazy.
Right. Yeah.
So how old is she?
She's 16 months.
Oh, very nice.
Her name's Hazel.
Oh.
Let's see. Let's see if I can find you. Cute one.
Oh, adorable.
He's bald-headed.
Oh, well, here's my our grandson. Yeah, in fact, we're going. Yeah, it'll be a year tomorrow.
Oh my gosh.
Yeah, so
it goes by so fast.
It does. Let me show you the picture here.
What's his name?
His name is Atreus.
Oh my goodness, that's so cute.
Here we go.
Is this your first?
It's our first, yeah.
Oh my goodness, he's so cute.
He is. That was in April.
Oh wow.
So he's even bigger now.
Oh, he's like so fast.
Oh yeah, I know. My son was telling me yesterday that He's just into everything.
Yeah.
You know, he likes to sneak up on the cats, so he'll just be a year.
Oh, like,
So,
is he walking? Or anything?
yeah, he is.
Hazel, she didn't start walking when she was like 14 months. It took her forever, but I live with my parents, so we have like 5 people like carrying her around all the time. So they were like, "Why does she even need to walk?"
Babies are smart.
Yeah,
If they can be carried, they'll
they're
they'll
like,
enjoy
"I'm gonna
that.
do this."
So
Yeah, but he's so cute.
so George. So George.
Yeah.
Yeah, there's a couple of them. Yeah, but you also starting. That's why she's done so. They've had the whole it's all of the Jewish background. A whole lot further. So she goes about this. Asian piece is moving, she's doing attention to secure it. But she can see the many things in her name. Bring there. Let's see.
Okay, let's move on and take a look at, we got through the four stages of the cardiac cycle. We finished up with isovolumetric relaxation. Couple terms here to remember. Touched on those the other day, and I was talking about blood pressure and mean arterial pressure and pulse pressure and things like that. Cardiac output. How much blood does your heart pump in 1 minute? The cardiac output is a measure of your heart rate times the volume of blood ejected with each contraction of the heart. So we use the term stroke volume. How much blood is being pumped at any given time? Now, when we say that the ventricles fill up to about 130, Yeah, here's our ventricle here. Here's the other ventricle here. 130 mils on this side, and 130 mils on this side. Here's the pulmonary trunk. And here is the aorta coming up behind that. So the body, so this is the pulmonary trunk, this is the aorta. Now, how much blood's going to get pumped out? Well, about almost half. If you have 130 mils at when the heart is filled, your stroke volume is typically about 60 to 70 mils. Maybe half what's there. The other half, or whatever's left behind, is the reserve. Because this is what we're doing right now. If you have 120 mils of blood coming in, you are gently pumping 60 mils out. We're not doing anything very strenuous right now. I'm not making you get up and run around or anything like that. So, but if we suddenly had this desire to exercise and started running up and down the hall, so we could irritate the chemistry class on the other side, not that we would, but if we did, we would have a demand for more oxygen. We have a demand to get rid of CO2. We have a demand for more glucose because we would be putting a greater strain on our muscles to do something. Our muscles require the removal of carbon dioxide. They require the addition of glucose. Our muscle cells require oxygen. So our heart would pick up the pace and pump faster to deliver more nutrients, glucose, to take away more waste products, and to remove carbon dioxide. And it immediately delivers some more oxygen too. So instead of during exercise, our stroke volume may go up to 80 or 90 mils, but we have that reserve. We have almost half of the volume in the ventricle after the contraction. After we contract, our stroke volume usually only takes into account about a half of the total volume. So if we have 130 mils in there at the point of contraction, we're probably going to pump about half of that out when we have our stroke volume. So our cardiac output is based on the number of heartbeats a minute. And let's say our normal heart rate is 70 beats per minute. And if our stroke volume were 70 mils, okay, here's heart rate. Here is our stroke volume, meaning amount per contraction. So if we have 70 beats per minute by 70 mils, that would give us a cardiac output. Yeah, 70 times 70 is 4,900 mils per minute. 70 beats a minute by 70 mils per contraction per beat gives us a total amount of blood coming out in 1 minute. The cardiac output would be, in this case, 4,900 mils of blood. Everybody see how I did that? Okay. So I don't like to do math in public, but I've done this before. So now this is our output, our cardiac output. How much blood are we sending out of our heart every minute? How much we have left in reserve? Well, probably another 4,900 mils, because we're only using half of the blood that's in the heart at any given time anyway. And we're not doing anything very, very, very demanding right now. So we're probably sending out 70 mils a contraction, and we're contracting 70 times. Well, our cardiac output is about 4,900 mils per minute, because none of what we're doing is very demanding right now, physically demanding. And so our cardiac output is simply based on stroke volume, which is the mils per stroke times the heart rate, the heartbeat, the pulse rate, whatever. And so we get, a nice round number of about 4,900 mils. Now here's a good example of it here. Stroke volume. The stroke volume is the amount, the difference between the end diastolic volume, meaning how much blood do we have in the heart before it contracts, at the end of diastole, at the end of the rest phase, when we have topped off the ventricle, when the atria have done their thing and topped off that last 20%, whatever is, that's the end of the rest phase. Whatever's in there at the end of the rest phase is what we call the end diastolic volume. Typically 120, 130 mils. The ventricles are filled up. What's left over after contraction is the end systolic volume. End systole at the end of systole, at the end of the contraction phase. What do we have left there? So if we pump out, if our total volume is 120 mils, and at the end of the contraction phase, at the end of systole, we have 50 mils left. That means our stroke volume is 70 mils. The end diastolic minus the end systolic. And so we see that, it can fluctuate all over the place. We're all going to be different. Anywhere from 60 to 80 mils is going to be per minute. It's going to be our stroke volume. We might find if we have a higher stroke volume, our heart rate is lower because we are accomplishing the same amount of perfusion with 50 beats a minute as opposed to 70. Perhaps here we have 70 times 70, we have 4,900 mils is our cardiac output. Suppose we had 50 beats a minute and we were putting out almost 100 mils per contraction. A person with a very healthy heart might be sending out more blood with less work. And so it just depends. But typically, stroke volume is going to be somewhere between 60 and 80 mils. The cardiac output is going to be somewhere between 4,000 and 6,000 mils per minute. We're all going to fluctuate, but that's how we are. Okay. And if you need another summary of stroke volume, it's again, stroke volume is the end diastolic volume at rest minus the end systolic volume after contraction. And it explains that again. So, a couple, good examples here. The heart rate, if your heart rate is 60 beats a minute and your stroke volume is 80 mils per beat, then your cardiac output is 4,800. So it's, for testing purposes, I try to keep it in nice round numbers so that it's easy to multiply. But it's going to change based on what we're doing. We're all going to be different. We're all going to have different stroke volumes and different cardiac outputs. It meets the demands of our individual bodies. So, what is the maximum amount that we can pump out of our hearts? Well, the maximum that's ever been measured is about 25 liters a minute. So, 4,900 mils is about 5 liters. So 5 times that would be the maximum that you could pump out. 25 liters per minute. Some people can reach 35 liters a minute. If you're a marathoner, if you are a sprinter in the Olympics coming up this summer, you have the different track and field, the different races. How fast are you running? What is your cardiac output there? 35 liters per minute, pretty amazing. So the reserve is simply the difference in here between our resting output and our maximum output. Now, most people have a reserve between, our maximum output is about 25 liters per minute, 20 liters a minute. And our resting output is about 5 liters per minute. So our reserve is the difference. We always have a reserve. A healthy individual will always have a reserve of blood in their heart because we don't typically only use about, at rest, we typically only use about half of our blood that's available. Now, the trick is, and I said this the other day in the Zoom lecture, that the stroke volumes have to be equal on both sides. What goes out of the left ventricle has to be matched by the right ventricle, because the left ventricle is sending blood to the body. And so it goes through the whole circulation of the body and then comes back to the right side. Remember, it's a closed system. What leaves the left side ultimately comes back to the right side. So if we pump blood out of the right ventricle out to, I'm sorry, if we pump blood from the left ventricle out to the body, and that blood comes back around, it's going to come back around and dump into the right ventricle. Everything going out the left side will end up in the right side. So if we have stroke volumes of 70 mils on the right and left side, the right ventricle sends 70 mils of blood to the lungs. 70 mils leaves the lungs and comes back to the left atria. Because that's where, blood from the right ventricle ultimately ends up in the left atria. The right ventricle goes to the lungs oxygenate the blood. That blood is returned to the left atria. So we send 70 mils of blood to the lungs, and we should have 70 mils going to the left atria. If we pump 70 mils out of the left ventricle, going out to the body, we should have 70 mils of blood coming back to the right atria. Whatever goes out on one side has to come back equal on the other side. Whatever leaves the right side of the heart has to come back to the left side with the same volume. Because if you don't, if you're sending 70 mils of blood to the lungs and the left ventricle can only pump 50 to the body, you're sending 70, but the left ventricle can only use 50. What happens to the other 20 mils of blood? You're sending every contraction of the right side is sending 70 mils of blood to the lungs to get oxygenated. Well, the left ventricle can only pump out 50, not 70. So you keep 70, 70, 70, 50, 50, 50 out. What happens to those 20 mils in each contraction? Where's it going to end up? It has to end up somewhere. You're going to see fluid accumulate in the lungs. You're going to have pulmonary edema because the left side of the heart isn't keeping up with what's coming in. Because you're sending 70 mils of blood to the left side of the heart, every contraction of the right ventricle goes into the lungs, comes back, but the left side is only sending 50 to the body. It backs up. So on the other side, if we have the left ventricle is pumping out 70 mils of blood just fine, it's going to the body, it goes, perfuses all the tissues, it drops off all the glucose, picks up all the CO2, and it returns back to the right side of the heart. And the right side of the heart has to send that blood to the lungs to become reoxygenated. Well, suppose that 70 mils coming into the right side of the heart, maybe the right ventricle can only pump 50 to the lungs. So 70 mils are coming into the right ventricle, 70 mils are coming into the right ventricle, and you pump, and you're only sending 50 to the lungs, and 50 to the lungs, and 50 to the lungs. What happens to the other 20 mils? It backs up into the tissues. And so your patient swells up. They have systemic edema. They have swelling in their arms and legs and their abdomen, because that fluid has to go somewhere. You have to have equal amounts. You have to have equal stroke volumes every time. Otherwise, that fluid got to go somewhere. It's not going to disappear. So you can't have 50 going out one side and 70 going out the other side. You got to have 70 and 70, or 50 and 50, or whatever it takes. So that is a critical point here. And when we have some sort of heart failure going on, depending on which side it is, left or right side, that's going to dictate where we would have fluid accumulation in the body. If the right side fails, we're going to have fluid accumulation in the tissues. If the left side is failing, we're going to have fluid accumulation in the lungs. Everybody clear in that? Did I over explain that? So, okay. Stroke volume has three things that affect it. What's known as preload, contractility, and afterload. Preload is what is the muscle tension in the heart right before contraction. Yeah. What is the muscle tension in the heart right before contraction? Contractility is what is the force at any given preload. And afterload, this is the easiest to understand. Afterload is the pressure on the other side of the semilunar valves, on the other, in the pulmonary trunk and in the aorta. What are the pressures on the other side that you have to overcome when you have to trying to force open the semilunar valve in the pulmonary trunk? How hard does the right ventricle have to squeeze to raise the pressure to get the blood to shoot through the pulmonary semilunar, just like the left ventricle is squeezing to send blood through the aortic semilunar? That's afterload. So preload. How much are our muscles stretched prior to contraction? And preload is actually not a bad thing. This cardiac muscle has sarcomeres. Everybody remember sarcomeres from AP1? Okay, just say yes, make me feel better. So, sarcomeres, the contracting unit of skeletal muscle. Well, cardiac muscle has sarcomeres too. That's where you have the A bands and the I bands and the thin filaments and the thick filaments sliding over each other. Everybody remembers that. If our sarcomeres in the heart stretch, if we have greater, if our cardiac muscle cells in the heart are stretched thanks to exercise, they're going to contract more forcibly, because when they're stretched, they have more binding sites for the myosin to bind to the actin. So a stretchy sarcomere in cardiac muscle means you can have more force in your contraction. So we have a more powerful contraction. Healthy hearts, exercised hearts tend to contract at more forcefully so that they don't have to contract as often, because they can pump more blood at a given contraction because they're stronger. They're stretchier. The more they can stretch out, the more their sarcomeres stretch out, the more binding sites you have to get more myosins to grab onto them and give them a yank. So you get a more powerful contraction. So that is preload. If our preload, if we have a good stretchy sarcomeres in our cardiac muscle, we're going to get a powerful stroke volume. We're going to have a very large stroke volume, but we may only have to have 50 beats a minute or even 40 beats a minute. Resting heart rate depends a lot on the elasticity, the stretchiness of the sarcomeres. So that is preload. Contractility is how much we increase the stretchiness, the strength of the contraction. How do we increase the contractive strength? Do we dump more epinephrine? Epinephrine increases the contractive strength. Epinephrine does that by opening up more calcium, calcium channels. Epinephrine allows more calcium to go into the cardiac muscle, which what calcium does, it binds to troponin and causes troponin to roll over and drag tropomyosin with it, exposing the binding sites on the actin for the myosin to grab onto. So contractility here, how do we increase our strength without stretching our sarcomeres? How do we increase the strength? Well, this is where we get into a fight or flight response. An individual in a fight or flight response is going to be surging epinephrine from the adrenal medulla. That epinephrine is going to be flowing into the heart. Of course, the cardiac nerve is going to be helping too. And so you have this epinephrine surge, and that's opening up these calcium channels and allowing more calcium to flood into the cardiac muscle, exposing binding sites, giving you a greater contractive strength. Now, it had nothing to do with a healthy heart, or an exercised heart. Lots of people go through fight or flight moments all the time. Every now and then you hear about somebody that picks a car off of somebody. You know, these are anecdotal sources of information, but it happens because, and they're not aware of it. They don't remember. They run through a burning building and pull people out of there, or well, maybe throw them back in. I don't know. Maybe they got confused. But they do remarkable and amazing things in a very short period of time using strength they weren't supposed to have. Their heart is beating so much faster, providing nutrients and oxygen, and taking away waste products from the muscle cells. And so you have this amazing strength temporarily. This is what contractility is. The influx of the sympathetic nervous system driving you to have greater strength simply because you're releasing more calcium into the heart. And so that, epinephrine is doing that for us. It has nothing to do with preload. It has nothing to do with how stretchy our sarcomeres are. This is an untrained individual. When I mean untrained, like an athlete, a trained athlete. This is just the average person having this sudden surge of epinephrine, releasing more calcium into their heart, so they get a more powerful contraction. All of these things can trigger an increase in thyroid hormone or glucagon. Glucagon triggers increased heart rate too. Epinephrine, digitalis, and high levels of calcium in the interstitial fluid. All of these are going to be drivers to increase contractility. This is not the difference between contractility and preload is that preload is a constant. An individual has worked hard to exercise their heart is conditioned, their sarcomeres are very stretchy, and they stay that way. So they have very powerful contractions constantly. Contractility is an event, and then another event as needed. It is not a permanent situation. It is driven by outside factors like epinephrine. It can also be blocked. Too much potassium interferes. Because, in this case, potassium, it's calcium and potassium, not sodium and potassium. So if we have too much potassium in the interstitial fluid, we're going to interfere with depolarization. If we're too acidic, we're going to interfere with depolarization. If your patient is taking calcium channel blockers to control hypertension, to lower their heart rate, then you're not going to see significant contractility taking place. Your patient's heart rate is going to be down. It's not going to be 70 beats a minute. It might be 60 beats a minute, and that's fine. It's controlling their hypertension. These contractility is affected by outside influences, whether for good, for increased contractility or reduced contractility. Increased contractility is going to be from epinephrine or excess calcium, for examples, or it's going to be decreased by too much potassium or taking calcium channel blockers. So contractility is dependent on outside influences. Preload depends on the stretchiness of the sarcomeres. That's an internal situation. Then afterload, essentially, what do you have to do to get the semilunar valves open? How much pressure do you have to raise to get the pulmonary trunk, the pulmonary semilunar, and the aortic semilunar to open. What's the back pressure? What's the pressure of the blood in the aorta pushing back against the semilunar valve? You know, you're trying to, you know, it's sort of like you're trying to go in the outdoor. You know, everybody's coming out and you're trying to go in. How hard do you have to work to push that door open to let you come in? Well, that's what's going on here with afterload. Afterload is the force we need to flip those semilunar valves open. So, is the pressure, if we have a patient with hypertension, they're going to have greater pressure in the aorta. They're going to have greater pressure in the pulmonary trunk. A person that has hypertension will have to, their heart will have to work even harder to get those valves to flip open. So the semilunar valve will only open if you squeeze the heart enough to overcome the back pressure. The same with the aortic semilunar. It will only open if you raise the pressure in the left ventricle high enough to overcome what's in the aorta. If you have a patient with hypertension and the aortic pressure is 180, for example, then you got to overcome that. You know, or the pulmonary trunk. If it's 40, which is not a good pressure, you have to overcome that. You know, so you have to, your heart has to work harder to deal with hypertension, which again complicates situations for the heart itself. Okay. Questions on any of those three? Outside factors that affect stroke volume. You know, we looked at preload, contractility, and afterload, but there are also outside factors that will affect the stroke volume itself. Acidosis, too much calcium, too much potassium, and the calcium channel blockers. Don't forget those. You know, I know I've said this already, but don't forget those. Now, what about the heart rate? How can we regulate our heart rate? Okay, so we have a patient that's bleeding out. And they're losing blood. So if they're losing blood, they're losing blood volume, then the heart's going to try to compensate by increasing the heart rate to try and maintain blood pressure. You try to maintain the heart rate and you try to maintain contractility so you can get those, you can get blood pumped to adequately perfuse the tissues. Now we can control that, or try to not control it, but try to regulate it through the autonomic nervous system. That's the big factor here, trying to regulate this. The autonomic nervous system will either release epinephrine or release acetylcholine to speed up or slow down the heart rate as needed. If we have a person bleeding out, they're going to try, and the autonomic nervous system doesn't know that they're bleeding out. All they know, all the autonomic nervous system knows, is that blood pressure is dropping because blood volume is dropping. So the signal to the heart is contract faster, more epinephrine. You know, that's the answer to the problem. Have the heart contract faster to make up for the lower pressure. The autonomic nervous system doesn't know that you're spewing blood out of an artery in a severed arm. You know, so they're going to, we get the complication out. The heart is beating faster. Yeah, and that's not a good thing there. Oh, what if you have a weak heart? Cardiac myopathy. The cardiac muscle tissue is weakened. You know, you try to overcome that. You try to stimulate the heart to contract at a faster rate. And of course, a weakened heart doesn't like to be told to beat faster. Often a weaker heart will respond to that kind of stimulus by enlarging. You know, an enlarged heart, like many other organs in our bodies, when they get larger like that, it's because they're less effective anymore. The thyroid gland gets bigger with a goiter, for example. And we see the heart doing that, enlarging. It isn't a good thing. So, what affects our heart rate? Well, a couple terms here. We refer to anything greater than 100 beats a minute as tachycardia. less than 60 is bradycardia on the average. However, many individuals have a resting heart rate less than 60 and are perfectly fine and are not in bradycardia. So before you take the pulse of an individual and determine that his or her heart rate is 55, you may want to ask a few questions before you start getting excited, because lots of people have a resting heart rate that low. A lower heart rate is caused by a couple of things here. The cardiac acceleratory center is inhibited. You're not dumping epinephrine into the heart as frequently as you need to. Maybe you don't need to. The cardiac acceleratory center is not as active. That's all we know. Or the cardiac inhibitory center is very active. Maybe you're getting more acetylcholine being dumped into the heart, slow the heart down. Again, we're going to work, you don't have to always have acetylcholine. You can simply depress the acceleratory center. Sedatives lower our heart rate. Different types of sedatives depress the central nervous system, so we're not going to be releasing as much epinephrine into the heart, for example. We can increase the heart rate if the acceleratory center becomes more active, or if the inhibitory center is less active. We can release epinephrine from the medulla of the adrenal medulla. Nicotine and caffeine all trigger an increased heart rate. They cause the heart rate to accelerate. A person who is hyperthyroid with too much thyroxine will also have an accelerated heart rate. They're sending out more energy. And what do you do with more energy? Well, more stimulus for energy. What do? Your heart beats faster. So our autonomic system is the regulator. It's not the controller. The autonomic system, you have both the parasympathetic and sympathetic side. We know that the sympathetic side gets us all excited and releases epinephrine. The parasympathetic side calms us down and releases acetylcholine. The sympathetic side gets turned on when we become stressed, or when we become excited, or we go into some sort of fight or flight response. We release norepinephrine. Norepinephrine it stimulates the SA node. It stimulates the AV node. It stimulates the muscle cells. And so we contract faster. Our heart rate goes up. Our end diastolic volume, how much blood we have accumulating in the heart at the beginning of the rest cycle, or at the end of the rest cycle, is less. Our heart is beating faster, so we have less time for blood to accumulate in the ventricles. So you're going into a fight or flight response, your heart rate picks up, and you're pumping blood faster. You're not doing 70 mils per contraction, you're doing 100 mils per contraction, or whatever. And since the heart rate has increased, there is less time for filling of the ventricles. So you don't have as much blood to send out. So you're sending out as much as you can. You're increasing your heart rate. Your stroke volume may drop. You are contracting faster and faster and faster, thanks to the presence of the epinephrine, not because of your sarcomeres, but because of the epinephrine. And at the other end of the time frame, the end systolic volume at the end of the contraction phase, you have even less blood left over. So if you're pumping out 100 mils of blood, okay, typically you would have 130 mils of blood at the end of the diastolic phase. You haven't contracted yet. And you would have about 70 mils of blood at the end of the systolic phase. You pump about 60 mils of blood out. Well, if your heart rate increases, that means there's less time for blood to accumulate. And so at the end of your diastolic phase, instead of 130 mils in your ventricle, you might only have 100. And you might be pumping out 90 mils of blood. And so what's left over at the end of the contraction phase, you may only have 10 mils of blood showing up in your ventricles, and then you're filling it up. What the heart will do, though, regardless of these numbers, what the heart will do is try to maintain a constant stroke volume. Doesn't matter what we have to manipulate, at the diastolic or the systolic phases of blood filling. We're going to send out as much blood as we can continuously. If it's 90 mils of blood per contraction, we're going to do that. Even if we resort to having only 10 mils of blood left in the ventricles, we are going to make sure the stroke volume is constant. We are meeting a demand. We got to go lift a car off of somebody or put a car back on top of somebody. I don't know. You have to do something extraordinary because it's a highly stressed, highly emotional, highly physical situation. And so your response is to increase the heart rate, maintain an elevated stroke volume. It doesn't matter how much you have at the end of the diastolic phase and how much you have left at the end of the systolic phase. If you're trying to get 90 mils of blood out in every contraction, you're going to do everything you can to do that. You're not going to think about it. We don't think about it. That's why it's the autonomic system. And I showed you this already. The two nerves controlling the heart, or rather regulating the heart activity. And we'll go back and forth on this. It depends on what we're doing. You walk up a flight of stairs, you might trigger more epinephrine. If you walk down a flight of stairs with less stress, you might trigger a little more acetylcholine. We're not aware of it. Our heart rate's going to fluctuate depending on what we're doing. We do know that our parasympathetic side is the dominant side. It always is. We're always parasympathetic dominant. That's our rest, digest, and defecate side. It always uses acetylcholine. Acetylcholine is going to hyperpolarize the SA node and the AV node by allowing more calcium to leave the cell. That hyperpolarizes the it opens, doesn't close. More potassium goes into the cell, driving its charge further away from threshold. So that's what acetylcholine does. It allows more potassium to come in. So your charge, your negative charge on the cells of the heart become more and more negative. So instead of being at minus 90 millivolts, it may hyperpolarize down to minus 160 millivolts, making it harder for it to reach threshold, making it harder for it to contract. That's what the parasympathetic side does for us. And typically, even at rest, the parasympathetic side is keeping our heart rate down. Without the release of acetylcholine into the SA node, our normal resting heart rate would be somewhere around 100 beats per minute. I mentioned that earlier. The parasympathetic system keeps our heart rate down to this 70, 75 mils per minute, 75 beats a minute. A couple things are going on here because we're not only just going to monitor, we're not going to just use acetylcholine and we're not going to use just epinephrine. We're going to be monitoring our own blood pressure. We have baroreceptors, pressure receptors here in the carotid sinus. You can find your carotid sinus, find your earlobe and drop down about an inch. And that's where the carotid pulse is located. That is where the carotid arteries split and go internal and external. So, and we have a very nice pulse rate that you can monitor right here if you very gently press against the carotid sinus. That's where they go. Two of the carotid arteries go internal and two stay external. We also have pressure receptors in the aortic arch. The heart is right here. Blood comes up, and in this area right about here are pressure receptors. And so we monitor the pressure in here. And if our pressure gets too high or too low, we can increase or decrease the cardiac output. We can increase or decrease the heart rate. If our pressure is too high, we can lower the heart rate so our pressure drops. If our pressure is too low, we can increase the heart rate to bring our pressure up. So the presence here of these pressure receptors is going to be telling the medulla, the cardiac acceleratory and inhibitory centers, raise the heart rate or lower the heart rate, because again, the vagus nerve and the cardiac nerve are going to be supplying information to the medulla oblongata. So, I mean, that vagus nerve is all over the place. And there's something here called the Bainbridge reflex. Has to do with filling of the atria. We know that we have the sympathetic and parasympathetic systems. They are antagonistic. Parasympathetic is rest and digest. Sympathetic is fight or flight. They are antagonistic. When one is active, the other is inhibited. You're not going to, now you're not going to have a sympathetic response and a parasympathetic response acting on the heart at the same time. So what will happen here is if we have a greater return of blood into the atria, this is where we get into the Bainbridge reflex. If we increase the blood flow to the atria will stretch. The atria will stretch out as more and more blood's coming in. And so when they stretch, that will stimulate the SA node. Yeah, because it's like a balloon. You're filling up the balloon with more and more water. It's getting bigger and bigger. It's stretching. Well, here, as more and more blood is coming into the atria, the atria is going to stretch. The atria can only hold so much. As it stretches, it's going to stimulate the SA node. The SA node is in the upper back wall of the right atria. Pressure of the blood in the right atria is going to tell the SA node, hey, we've got more blood here. And so we will increase the heart rate in response to help get rid of this extra blood. We also see the stretching is going to turn on the sympathetic system to release more epinephrine. The bottom line here is very simple. Atrial filling leads to a higher heart rate. More blood coming into the atria increases the heart rate. That's what happens. Our heart rate goes up as we have more blood coming in. If we have more blood volume, then we have a higher heart rate. If we have a higher heart rate, we would have higher blood pressure. Now consider what happens when you have a patient that is consuming lots of salt and retaining lots of water. Remember that water tends to follow salt. So a person that has a lot of salt in their diet is going to retain a lot of water. Where's that water going to end up?
Their tissues.
Hmm?
Their tissues.
In your tissue, yes. Well, before that's going to be in your blood. Remember, everything we eat and drink ends up in our blood, and then it's going to go into our tissue, and we're going to swell up. But before that happens, your heart's going to have to beat faster, because the more fluid you retain, the more your heart has to beat. So, what do we tell hypertensive patients not to eat a lot of? Salt. Thank you. Yes, salt. Don't eat a lot of salt, because if you eat a lot of salt, you're going to retain water. And if you retain water, you're going to increase your blood volume, which is going to cause your heart to work faster and harder. And you increase the blood volume, the atria are going to snitch on you and say, hey, we've got too much blood volume here. And then they're going to tell the heart to beat faster. Then you're going to end up with all that extra fluid. It's going to end up in your tissues, and you're going to be saltier, and you're going to be thirsty because you're eating all the extra salt. It's it's what our body does. So we try to advise hypertensive patients, try to avoid the salt. It's almost impossible to avoid salt these days. I mean, everything, even anything, any prepackaged, even canned foods, have some small amount of salt in there that you just can't get away from. So, okay. Let's see. Okay, what else controls the heart rate? Well, of course, epinephrine and stimulates contraction. Thyroxine, thyroid hormone, increases the heart rate. So, sodium and potassium and calcium ions affect the heart rate. Calcium levels go up. Sodium's going to tell you to retain water, which will indirectly increase your heart rate. Calcium levels will affect the heart rate, because if you have more calcium or less calcium in the interstitial fluid, you'll have either more or less calcium going into the muscle cells when they depolarize. Too much potassium or too little potassium interferes with repolarization. There are lots of things that can go wrong. A person who has a major dose of potassium, now we all know potassium is good for it. Who hasn't had muscle cramps, right? Who hasn't eaten a banana? You get a little potassium if they have a muscle cramp. Maybe you don't like bananas. I don't know. Remember, don't let monkeys eat bananas. Couldn't resist that. So, anyway, excessive amounts of potassium are going to hyperpolarize your cardiac muscle cells, making them harder to hit threshold, and the heart's going to stop. People can die, from potassium overdose. Got to be a lot of potassium, but it's entirely possible because it causes the heart to hyperpolarize. It can't get back to threshold. It's a muscle. It acts just like any other muscle does. So, a couple things that can go wrong with calcium. Too little calcium will lower the heart rate. People that take calcium channel blockers, for example, will have a reduced heart rate simply to lower their blood pressure. It's a very effective hypertensive medication. Too much calcium can increase the contractility. We have more calcium in the interstitial fluid than we need, and it's going to be depolarizing the cell. It's going to make it harder for the cell to repolarize. Too much potassium can lead to cardiac arrest, again, because it's going to drive us further away from threshold. And not enough potassium will weaken the heart rate. We still have to have a certain amount of potassium for the muscle cell to repolarize. So, okay, I have 11:27. Let's stop here, and I will continue this in Zoom land tomorrow. And